Hi there, and welcome to Algernon Pharmaceuticals Facebook Live Update. I'm Cindy Edwards from PR Media Now, and joining us today are Chris Moreau, CEO of Algernon Pharmaceuticals, and Dr. Christopher Bryan, VP of Research and Operations at Algernon. Now, Chris, on Wednesday, your top-line data came out on Algernon's COVID-19 Phase 2b/3 trial with Ifenprodil. Can you walk us through that?
Sure I can, and hope you had a good weekend, and welcome to everybody joining us today. So on Wednesday, we announced top-line data, and what we announced was that we saw basically in the 20 milligram dose of Ifenprodil, three endpoints where we saw activity. The first was in all-cause mortality. We had 0% mortality at day 15 in the 20 milligram dose group. And we had achieved a p-value of 0.16. So we didn't reach statistical significance, which is 0.05. And that is not totally unexpected since the study was a pilot study of only 150 patients. So we expected a lower N in terms of mortality. Of 150 hospitalized COVID patients, you can expect about 15% to go on to mechanical ventilation, including non-invasive, which would be something similar to a CPAP machine or air-assisted, and then a percentage would go on to invasive mechanical ventilation, where you're actually put out and they have to put tubes into your lungs to help you breathe. You're looking at about 6% mortality. So we saw that signal at day 15. That's important because we only treated folks for 2 weeks. There was an increase in a person we had mortality in the 20 milligram group between day 15 and day 29, which might suggest that patients would have done better, or that patient, if they had stayed on low dose, that's a possibility, the low dose Ifenprodil. The second area that we saw activity was in return to normal oxygenation. We were very close to statistically significant at 0.06. We had 100% of patients on the low dose group of Ifenprodil achieve normal oxygen at 4 days compared to 9 days for patients in the untreated arm.
That seems significant to me.
It's very significant. It's a significant endpoint. It's an approvable endpoint based on the data and the guide from the US FDA. We still have yet to communicate with the FDA. We're working on that communication, and it's part of our completing our review and speaking to them about the data. It is significant, and it also reached, it was very close to statistical significance. The third area was overall time in ICU. And we reported a p-value of 0.0315. We put up a caution that there was a flag from our statistical review group that they're still trying to work through. So we did report the data, but we said there's a caution, there's an anomaly in the data. And this data showed that there were less days spent in the ICU for patients in the 20 milligram group compared to the untreated arm. Now, we've had lots of feedback. I've been speaking with some of our investor funds, institutions, analysts. The reason we were conservative in terms of our description when we put out that data, we really just put the data out without referring to it in any way other than to say, look, this is the data and we're working through it, is we didn't see a signal we may have been expecting in the 40 milligram group. So to remind everybody, we had 3 treatment arms. We had untreated 50 patients. They got standard of care. We had 20 milligrams. They got standard of care plus Ifenprodil. Then you had a 40 milligram treatment arm. So they've got a higher dose of Ifenprodil plus standard of care.
Well, I was going to talk to Dr. Bryan about that 40 milligram group. Let's talk a little bit about that. Did you or did you not get the results you were expecting with the 40 milligram group? And how do you explain the results that came out of that?
Certainly, Cindy. So just so everyone is aware, the 20 milligrams 3 times daily dosing is the on-label dosing in the jurisdictions where the drug is approved for other indications. And that was what we'd intended to run with. The FDA actually suggested us we ought to consider a higher dose, thinking that there may be a benefit with that. And we should clarify, for this indication, we didn't have the sort of preclinical standard background that one would want to typically move into a phase 2. Just because of COVID, because of the urgency of the situation, we chose to run based on predominantly on data which was not ours, on this H5N1 mouse study that Chris has brought up previously. And so we don't really have a rock solid, I would say, understanding of some of the pharmacology and the dose effects, which I think is what is playing a role in this situation, which has happened with the 40 milligram. So Chris mentioned the 3 positive trending signals that we saw in the 20. Those signals were not present in the 40 milligram. And so it's important, I think, to look at the whole of the data and not just focus on one particular piece, positive or negative, that might lead us astray. And so it's essential that we understand why the 40 milligrams may not be having that positive clinical outcome. Now, it is known that a lot of receptors, you can overwhelm them. So you can imagine a couple possibilities. One would be you reach a threshold and then you have no further effect. But what may be happening here is that overloading the receptor may actually be leading to some sort of negative effect and leading to a situation where our high-dose treatment group is indistinguishable from the standard of care. And so we have some plans to very quickly potentially conduct our own research, as well as speaking to some experts in the field, and to try to see if we can produce quickly an answer that gives us some confidence as to the cause of the effect we're seeing.
Okay, now, Chris, to run a study on mortality to see if the 20 milligrams of Ifenprodil really works, you only need 1,900 patients in your phase 3. So that seems reasonable. Other trials have had upwards of 5,000. So can you get this 1,900, and what's the cost to go down that route?
So it's not a massive study. I mean, hydrochloroquine, remdesivir, dexamethasone, these were 5,000 plus patient studies. It's interesting, while it's 1,900, so we projected, and part of that phase 2B objective was to not only find a signal, but then to do a calculation and find out how powered would a phase 3 need to be. So for mortality it was 1,900, but for oxygenation it's only 450. So that's a smaller size of a study, and that's sort of more in line with our cost projections from what we were initially. I think we had sort of estimated 11 to 1,200 patients would have been 10 million plus. And part of this, what we're doing behind the scenes is sort of figuring out, and it will form part of our communication with the FDA, confirming what is an approvable endpoint. We will need a placebo. You may not need a placebo in a mortality study, but that's 1,900 patients. This will add a bit of time and cost to get prepared for a phase 3 study if that's the best plan forward to move it ahead. And then what's the backdrop as well of what's going on in the market in terms of all of the vaccinations, 100 million doses in the US, 200 million to be delivered in the next month or two, approved everyone over the age of 18. The smaller study, the better, because what if maybe the severity of COVID still remains, but the number of people getting sick or less could be tougher to run that study. And so these are all part of the considerations that we're looking at as we're looking at the data and trying to figure out our next steps.
Dr. Bryan, is there a timeline for when you would start a phase 3 if it looks like that's the way you're going?
Well, it does take a little bit of time to get things up and running. The adaptive design that we had here, the 2B/3, would speed things along, but I would expect you'd be looking at least a couple of months before you'd be back into patients again, just because of all of the paperwork and approvals that are required. If we do choose to manufacture a placebo, it would be sort of along those timelines as well, 2 to 3 months to be having that on.
Now, Chris, what are the next steps then for Ifenprodil and Algernon? Are you going to talk to the US FDA about this?
Our plan is to communicate with the US FDA. Right now, behind the scenes, we're completing the review of all of the data. We are reaching out to a couple of other research scientists, one in particular from Japan who has an expertise in NMDA receptors and has worked with Ifenprodil. We were searching the literature to see if there is a class effect. Memantine, ketamine, these are NMDA receptors. They seem to show that they can oversaturate the receptor and you can get sort of a bell curve shape response. So like in entertainment, less is more, and that may be the case with Ifenprodil. We want to have that information, and then the next step to communicate with the FDA, get some feedback on sort of next steps, and then depending on which way we're going forward, what is the cost to do our phase 3 and what type of support, if we firmly believe that the study justifies support, that can come in from various places. Could it be a research grant, research partner, commercial partner? We're already in a difficult situation with a repurposed drug because it's a bit different than an NCE. And so lots going on behind the scenes. We wanted to reach out. I think the market was wanting to get some answers. We talk about in these sessions or others more about what has been announced as opposed to what our plans are. And we don't have material disclosures in these discussions. They're just to give some color and insight into what we've announced.
We've got a few questions coming in from our viewers. Jamie Gregory asks, are there any chances of any government or Gates Foundation support?
There may be. We were working with the Gates Foundation on a prophylactic study application when, and we had advanced it quite far. Bryan was working with us and was running that or managing that contact. And they simply got back to us and said, we're sort of shutting down. We've run out of funds. So could we sort of go back to them and go back with this data and say, we believe we have a reasonable scientific argument on why we didn't. And in fact, I just want to add, there is some trending negative data in the 40 milligram. Trending negative data, not statistically significant. So that would also suggest that in fact too much isn't helpful, and in fact it has a negative effect. But you can't ignore this mortality signal. I mean, in the dexamethasone study, they had 3% reduction in mortality, and that's about what 3.3%, along with normalization of room oxygen and days in ICU. This is so far it's compelling, but we have to complete our work, our research in them, and understand what's going on.
Another question for Dr. Bryan: is it possible for Ifenprodil to be working but not showing signals on the news score, length of hospitalization?
I think it is. I think it's easier to understand maybe if you think about going down to the patient level. So we talked about perhaps seeing a signal on mortality. You could imagine if a patient does pass away, they'd be out of the hospital potentially very quickly. So in some cases, a longer hospital stay may not be a bad thing, and that may be what's showing up in these sort of confounding signals.
We have another question here.
Cindy, can I add?
Sure, go ahead, Chris.
There's been lots of discussion about the news score and WHO score. The news score, think of it like the Dow Jones. It's a composite index of a wide grouping. So with the news or WHO score, it's a average score of a number of different markers and results. However, we went after the sickest of the sick with Ifenprodil. So we went after hospitalized on oxygen. And if you're going to see the signal, you're going to see it in people who are getting worse. And keep in mind, 85% of hospitalized COVID patients get better. So it's really tough to show a delta between the treatment arm and the untreated arm because most people are getting better. And so with the WHO score, which is an overall, if we're really impacting the smallest number of people, 15% of the people in the study, it's not it makes sense to think that we're not going to be affecting the WHO score because the majority of people are getting better. And it's the reason the FDA came out after we had filed that adaptive design and said, we're not keen on the WHO or new score. Approvable endpoints are mortality, days in ICU, days in hospital, number of patients on ventilation, returning to normal oxygenation, not WHO or news.
What about the EUA?
I mean, we will speak to the FDA as part of our communication. Challenging. I think we'd have a much better shot had we achieved statistical significance in mortality. That was really or closer to it. And again, it's in the backdrop of what's going on, and it's always situational. So you've got, let's say that it was different. No vaccines. We're in our 6th lockdown. No other therapeutic is working. Ifenprodil is a very safe drug that's been proven. I think you would have a much better audience to get an emergency use approval. But with this data is really, I think, a stronger argument if we're satisfied to do that for a phase 3. And I think the likelihood of EUA is small based on what we're looking at right now.
There's a follow-up question from Raphael saying, what about lung scarring?
So it's very tricky because, to remind people to go back, we originally were investigating Ifenprodil for idiopathic pulmonary fibrosis. Idiopathic means of an unknown origin. If you have fibrosis from COVID, that's no longer idiopathic. It's COVID-related fibrosis. And we really, our data for IPF and chronic cough was our data. We own it. We ran what's known as a bleomycin model for mice, where we insulted the lungs of these mice with bleomycin. 7 days later, we started to treat with Ifenprodil, so the lungs already start to scar. So you have scarring. We treat with Ifenprodil. We use comparator arms of nintedanib and pirfenidone. This is Boehringer Ingelheim and Roche's drug for IPF, and we outperformed them in reducing the fibrosis. We also went against Merck on cough, and we saw a very positive effect. Now comes COVID. The H5N1 study, they had pre-treated the mice by 1 day, and they had this massive reduction in mortality, 40%, reduction in acute lung injury, reduction in edema, cytokine storm. It was a reasonable argument to make that it might help with COVID patients. But we're asking 2 different pharmacological endeavors for Ifenprodil. One is, can it reduce fibrosis? Or 2, can it reduce the insult while it's happening? And, right now, I don't think we have enough information. I know there was some information that came out that suggested patients were doing better on their exit, but it wasn't really part of our study. And that information is there for us, but it could also be confusing because patients leaving who are either coming back or having issues also are having heart issues, liver, kidneys. And so I think we need to continue to do our data review. We don't really have an answer yet on this issue of lung scarring. Is it the problem that, and I'm just not talking about hearing a media report, it's on CNN or a local paper that so-and-so had COVID and now they're struggling. We're really talking about a larger part of the patient population who are suffering, and is it fibrosis? And then would Ifenprodil assist? Those are some of the things we need to work through.
I'm seeing a lot of questions asking the same type of questions. What about the fibrosis endpoint? When will we see the IPF results? Why is the IPF sign up so slow? What about the chronic cough results? When are these results going to be made public?
Chris, do you want to chat a bit about the study?
Sure. I think the main problem with the IPF study is the that COVID has made people very reluctant to participate. That's one big factor. We're doing that study, as people know, I think in Australia. We've just added a couple of sites in New Zealand as well, trying to do anything we can to accelerate the pace of the enrollment there. It's quite a competitive space in terms of recruitment. So there are a lot of patients we're waiting on who are either they participated in another study and we're waiting for them to become eligible for ours, but there are also patients who, we have certain physicians who are investigators for us, and they've looked at their entire pool of patients that they treat and reached the end. So we've actively begun to explore other ways of bringing patients on board. We've looked at various recruitment campaigns, social media, some organizations that seem to be just perhaps on the verge of bearing fruit for us. So I hope to have a more positive update on that study within the coming week.
Chris, as you know, everybody wants information yesterday, and I am seeing that from a lot of our viewers saying, so give us a time frame. When are we going to get notification of the plans? Do you expect we'll get notified in the next week or month? Give us an idea, please.
Yeah, I don't think I think we're talking days. We're not talking weeks or months. And just to go back on the IPF, we on our IPF and cough, I hope it does make intuitive sense that as a company and management, we want to get that study fully enrolled as soon as we can so that we can get that data. We can't move forward without it. The management team isn't flying to Sydney, Australia, trying to convince patients as they come in to their clinics to enroll in the study. We've hired Asia Pacific's top CRO, Novatech. They all of this tremendous amount of work ahead of time. They identify PIs. They sign up, there's ethics approval, then they go through their patient population. We a lot of this is out of your control what the velocity is. This is also a orphan indication. These are very sick folks. There's a benefit to Australia. We get 40% of our funds back, but it's a smaller market. So we have restrictions also. What would be nice is if we could add a couple of different countries, Hong Kong, South Korea.
That goes along with Major's question here. Why not spend the money and get patients from another place?
Because it's the challenges with Ifenprodil. We really had to keep our costs down. And this will give folks a little bit of background on the Algernon super cost-effective repurposing model is to not have to manufacture your own drug. So we were able to isolate a supply of Ifenprodil from a Japanese supplier, export it into Australia. It was very difficult. And the reason it's so difficult is because the manufacturer in Japan doesn't really want you doing any studies with their drug, because if there's any sort of adverse events, they have to report it. It could interfere with their market and what they're doing. And they don't have any control on the study. So we were quite lucky, Algernon, to get access to Ifenprodil, to get it into Australia. It was a very, very tough go. The alternative would have been we could have still done the study, but we would have had to have made our own Ifenprodil. That would have taken 6, 7, 8 months. And we started to manufacture our own supply for an intravenous application if needed for COVID. So we're restricted right now to Australia and New Zealand. We can't change up our supply because it will change the study. But I just hope folks really understand that we want the study, we want full enrollment just like we did with COVID as soon as we can. There are some limitations on what we can do, but we're doing what we can to make sure that it's fully enrolled and we can get access to the data.
Okay, here's a question from Alex. Dr. Bryan, does the positive signal for the oxygen saturation translate to positive results on the IPF study, or can it?
Well, certainly you'd rather see it trending that way than the other way, but I would be very cautious about trying to extrapolate from the COVID study onto the IPF cough study. As Chris has said, we're talking about different pathologies of disease, and I would say we have a much firmer belief in no, that's not quite right. Are the studies that we ran that underpin our decision to begin that IPF chronic cough study are really quite solid. in comparison to what we had to do for COVID. I think we should be more reliant perhaps on our preclinical data there than any signals that are suggested by the COVID study.
Chris Moreau, I'm going to let you have the last word here. I think just overwhelmingly from the comments we're getting, I mean, people are trying to stay positive, but I think they're getting restless. And I know that you hear that all the time. So I'm going to let you have the last word here.
Yeah, I just want to make a comment, and I'll come back to a summary on when we found out about the H5N1 study for Ifenprodil in China. Mark Williams and I worked quite hard to find an animal study for COVID, but they didn't exist. This was last March when we started, keep in mind. And Mark actually reached out to one of his former professors at a university in Alberta, who was actually funded by the Canadian government. They had isolated COVID and were working on an assay, but we just couldn't get access to it. So we really had to move forward just based on that H5N1 study. Later on, UT Dallas did confirm that there was an overexpression of the gene responsible for the NMDA receptor, 600-fold overexpression, when they looked at lungs from healthy patients compared to COVID-infected patients. That gave us some comfort we were on the right path. But so we've done our best with the data that we had. And I totally get investors wanting to have data ASAP. There's no need to explain that. That makes sense. We're working as hard as we can, and not only, of course, on COVID, but we're working on IPF cough. We've also been advancing a DMT program for stroke. And so we've got lots on the go, and just encourage investors to continue to be supportive if they like the story and if they like the progress. And I mean, that's a personal decision, but we're continuing to advance, and we're confident of the work that we're doing.
All right, Chris Moreau and Dr. Christopher Bryan from Algernon Pharmaceuticals. Thank you both very much for joining us today, and to everyone who tuned in. We appreciate you, and we appreciate your questions as well. And of course, you can always tune in to Facebook, Algernon's Facebook, for Facebook Live updates, and algernonpharmaceuticals.com for more information. Oh, we got one more question that came in right before I let you go. There's some. Oh, never mind. It's too involved. You'd have to open up the article and then you'd say. Natalie asks any thoughts on this newly released article. Well, we haven't had a chance to read the article, so we can't comment on that right now. But maybe next time. Thank you very much for joining us today. Have a great day, everyone. We'll see you next time.
Thanks so much. We'll be in touch soon. Take care.
Okay. Thank you.